Hello guys, I am Yatanjeet Kaur and welcome back to my channel. Today I am making a video on the neuroanatomy. So let's discuss about the neuroanatomy. Generally, our brain is divided into the three parts: the forebrain, the midbrain and the hindbrain. The forebrain is also known as what? It is known as prosencephalon, and midbrain is known as mesencephalon. The hindbrain is known as rhombencephalon. Now, as you can see, as there is a formation of these three structure, these spaces are known as the cavities, and these outside structure that are in black line, that are walls. So the cavities and walls further give rise to certain structure that we will discuss on later. Now, the prosencephalon is further divided into the two parts. The first one is the telencephalon. This is the telencephalon. And the other one is what? The diencephalon. Basically, the telen and the diencephalon, they will form the walls of these structures. And the midbrain, it will not divide to any structure, it will remain as this mesencephalon. Now the rhombencephalon will further divide it into two parts, that is metencephalon and the myelencephalon. Now this meten and myelin, as I told you, they will form the walls of the structure. So what walls they are going to form? The telencephalon will form the cerebral hemisphere, plus the basal ganglia. And the diencephalon will form the thalamus and the retina. And the metencephalon will form what? As I told you, midbrain will not form any wall of any structure because it is not dividing. So the metencephalon will form what? It will form the pons and the cerebellum. Pons and cerebellum. And myelin means soft, it will form what? Medulla oblongata. So basically you can say the hindbrain will give rise to what? The brainstem structure, the pons, cerebellum and the medulla oblongata. So as I told you, they also form some cavities. So what cavities? Generally, these cavities are you seeing here? It will give rise to, generally here the here the lateral ventricles are located. In the cavities of telencephalon, you will see the lateral ventricle, okay? And in the cavity of the diencephalon, you are going to see the third ventricle. And the cavity of the mesencephalon, here you will see the cerebral aqueduct. What you are going to see here? Cerebral aqueduct or duct of Sylvius. Now the fourth ventricle will locate where? In the cavity of the meten and the myelin one. Generally, the upper boundary of the fourth ventricle is formed by the metencephalon, whereas the lower boundary of the fourth ventricle will be formed by the myelencephalon. So guys, this is all about the neuroanatomy of the brain, different parts. Now we will focus on what? About the function of the brainstem structure. Now about the function of the brainstem structure, like pons. So what is a function of pons? Generally, the pons is responsible for the waking and the relaxation. So how you can remember this? Just remember a simple mnemonic, that is pons. Just remember this as with the day. So when you see the pons, what you will feel? Very relaxed, very soothing feeling. So that is mainly responsible, which structure is responsible? That is pons. For relaxing plus waking. Okay, now the cerebellum. Now to memorize this function of this, it is mainly responsible for what? The coordinated movements and the balance. For coordination and balance. Coordinating movement and balance. So how you can memorize? I will draw just a figure. For example, this is a rope. These are the two poles. This is a rope and the one person is standing over this pole with the two bells. This is one bell and this is the another bell. So just remember, the walking and the balance is maintained of this person is maintained by what? By the cerebellum. By seeing this picture, you will memorize the function of the cerebellum. Now the medulla oblongata. The medulla oblongata, what is its function? It is mainly regulate the heartbeat and breathing. Okay. So how you can remember this? Just picturize an athlete who is wearing a gold medal, and that gold medal is covering the athlete's heart as well as the lungs. So you can remember, जब वो भागा होगा, उसको सांस चढ़ी होगी, heart beat फूली होगी, और जो उसका gold medal है, will just pinpoint you where you have to, what things the medulla oblongata is coordinating. So just remember the example of athlete with gold medal covering the heart and the chest and the lungs. So these are the functions. Now we will discuss about the blood-brain barriers. Now, blood-brain barriers. Generally, the blood-brain barriers are mainly formed by the three structures. So, I will draw the diagram. This is the endothelial cells. These are the capillaries. Okay, these are the endothelial cells. And here you will see one structure that are astrocytes. These are the astrocytes. So what are the three structures that will form the blood-brain barrier? One is the astrocytes, the second one is the basement of the basement membrane of the capillary endothelium, and the third is the tight junctions. So these are the three structures that are mainly responsible for forming the blood-brain barrier. Okay, so the most important among three is one, that is the tight junction. So if the question comes to you, what is the true blood-brain barrier, your answer must be what? The tight junctions. Okay, so tight junctions are the main blood-brain barrier. Now, in some cases, like in tumors or in infections, the blood-brain barrier can be destroyed, and they can lead to what? They can lead to the vasogenic edema. Now, what is this vasogenic edema? In this, the fluid from the capillaries will come into the extracellular spaces, and there will be mainly they will affect the what? The white matter. Okay, so just remember destruction of blood-brain barrier can leads to what? The vasogenic edema. Now in brain there are certain spaces where you can't find the blood-brain barrier. In general, the blood-brain barrier is there around the ventricles, and those spaces where you can't see the blood-brain barrier, those spaces are what? Example of those spaces where blood-brain barriers are absent, those are what? Area postrema. I will tell you what is it. Subphonical area, region, neurohypophysis, and the pineal gland. Okay, now this area postrema, it is also known as the city Z zone, and it is a vomiting vomiting center. So mainly in the medulla oblongata, the area mainly responsible for the vomiting reflex, that is the area postrema. Just remember this. So this is about the blood-brain barrier. Now we are going to discuss about the arachnoidal spaces. For that I have to draw one diagram. For arachnoidal spaces, just remember this is a dura mater. This is the meningeal layer of the dura mater. This is what? Meningeal layer of dura mater, and here you have certain villi like structure. This is what? Arachnoid villi, noidal villi. Now what is happening in this is what? Arachnoidal spaces. Okay, so what is mainly happening here? This is all this area. This is what? This is the dural venous sinus. Okay, so what is mainly happening here? The arachnoidal villi will help for the reabsorption of the CSF. So mainly the CSF will go through the arachnoidal villi into the DVAs. Okay, so if they ask you what is the function of arachnoidal villi, it is generally the absorption of CSF. Now sometimes what is happening when arachnoidal villi are like this, when they get old, they will form something called arachnoidal granulations. What? They will form arachnoidal granulations. Now what you are going to see, there will be the granule formation like this. So these are the arachnoidal granulations. When these arachnoidal villi get old, they will form these granulations. Now these granulations mainly seen over the superior sagittal sinus. Okay, so where they are mainly present? On the superior sagittal sinus. Now more important point about these are, generally the mixed or there is any angioma of the subarachnoidal space, it is also located where? It will locate over the superior sagittal sinus. And if they ask you the maximum reabsorption of the CSF, you will see where? You will see the maximum absorption of the CSF over the superior sagittal sinus, because here there are more arachnoidal villi, and what the function of these villi? For the absorption of CSF. So indirectly you will see what? More CSF absorption over here. Now after that, after the arachnoidal space, we will discuss about the subarachnoidal spaces. Now subarachnoidal spaces. So I have to draw one diagram. So this is the corpus callosum, okay, and here you will see the medulla, the bones like this, and here the cerebellum behind the bones. So it is like this, and it is covered by the pia mater. So in subarachnoidal space, there are mainly, you will see all the large vessels, the cranial nerves over where? The subarachnoidal space. Generally the subarachnoidal space is comprised of the 5 major systems. So we will discuss about them one by one. Okay. So these space that are you seeing here, this space is nothing but it is known as the cisterna magna. Now what is happening in this cisterna magna? It is also known as what? Cerebral medullary cisternae. And as I told you, in subarachnoidal spaces you will see certain large vessels, the cranial nerves and the CSF. So here you will see which vessel? You will see pica, that is posterior inferior cerebral artery. Plus you are going to see the 9th, 10th and 11th cranial nerve. So 9th, 10th and 11th cranial nerves plus what? CSF. So how I remember is like 9th, 10th, 11th are the greater number. So cisterna magna is also big. So just remember like which cranial nerves are present, 9th, 10th and 11th. Now the space you are seeing here, this space, this is also one cisterna, that is superior cisternae, or it is also known as the cisternal ambience. Cisternae ambience. So what you are going to see here, you will see one vein, that is vein of Gala, which is also known as the greater cerebral vein. Here you will going to see what? Vein of Galen. Now here you will see what? Interpeduncular cisternae, and this is in front of the pons. You're going to see the cisternae montes. Okay, Here you will see what? Cisternae pons. Now what are the structures present in them? We will discuss. In interpeduncular cisterna, why it is known as the interpeduncular cisterna? Because it is present between the two peduncles. So it is known as the interpeduncular cisterna. Generally in interpeduncular cisterna, the main content is what? The circle of villi. So you will see what? Circle of villi is the main content of this. Plus the third cranial nerve is also present in this. Now cisterna pons, here you will going to see the basilar artery plus the origin of anterior inferior cerebral artery and the sixth cranial nerve. So these four things you are going to see in the subarachnoidal space, and particular large arteries and all the cranial nerves are located mainly here. Now in this one more thing, like if this is a brain and this is our sulcus of Sylvius, then you will see the space here, the cisterna over the sulcus of the Sylvius. You will see the middle cerebral artery. You will see middle cerebral artery over here. So guys, this is all about the neuroanatomy. In next video I will make the spinal cord and the tracts also. So please subscribe my channel and please hit the like button, share my video. Thanks for watching.